kids. Today we are going to talk about transcription in prokaryotes. So before you start the topic, we have to understand very few basic things about the structure of DNA and RNA. So DNA is a macromolecule, it's a genetic molecule. It has the genetic information stored and it has exactly two opposite strands. It has a double helical strand and the two strands exactly go anti-parallelly. That means their polarity, they are opposite in direction, but they shares exactly the parallelism also. In DNA, what we can find that there are double helical structure. So double helical structure, it is, what did I tell? Right now only I have mentioned DNA has anti-parallel double strands. So one is 5' to 3', that means another is again 3' to 5'. So this is the direction. And what are the bases, nitrogenous bases we can find here? These are adenine, guanine, cytosine and thymine. In case of RNA, we can find again here only single strand and the bases are present. Those are adenine, guanine, cytosine and uracil. We cannot find thymine in case of RNA. Actually, it is thought, it is hypothetically it is believed that from RNA only the DNA is evolved, and RNA, the uracil is very unstable. So to make the genetic information more stable, here we got actually 5' methylation in the uracil. The thymine is actually 5' methylated uracil. So we can understand, if I need to make from DNA to RNA, my thymine will not be present. Here obviously we can get the uracil. And remember, what is transcription? The process of making the RNA from DNA. This is called as transcription. Now why we need to understand this process? Why to understand this transcription process? Actually, we all are made up of the single cell. Now from one single cell, how a multicellular big organism like an elephant or blue whale or human being is made up of, so we have to understand that procedures. That how a single cell has the all information how the organism is going to be look like. We all know for sexual reproduction, if we talk about the example, this is a zygote, this is a single cell, we all are made up of this, and from this zygote, exactly a fish, from a zygote of a fish is completely built. How this is possible? Because from DNA, RNA will be produced. From RNA, there will be another process which is called as translation. From RNA here, we will be getting the protein. We all are made up of proteins. We all are. My hair, texture, my skin, my nails, everything, whatever I have, the features, it is only for the proteins I have in my body. So the protein making or protein formation process is very important. From DNA directly we cannot produce the proteins. For that we have to one step ahead, that is formation of RNA, messenger RNA, and this process is called as transcription. So here what we have understood, what is transcription? The process of formation of mRNA or messenger RNA from DNA, and what you have understood, the DNA structure, the bases present in the DNA and the bases present in RNA. Now exactly I am going to talk about the transcription process. Now here what we can find? We can find that I am going to only talk about the transcription in prokaryotes. I am not going to talk much in this video about the transcription of eukaryotes because there are some post-transcriptional changes also. But here in this video, I am only going to talk about the process possible in case of prokaryotes. What are prokaryotes? We all know they have genetic information, their structure, their cellular structure is a little bit different from us. They have polycystonic genes and true nucleus is absent. Okay, now let's check it out how the transcription takes place. Okay, so transcription, if we talk about the transcription, obviously we have to know about transcription. transcription unit. That means what are the components which are going to be involved, or what are the machineries we are going to use in this process. So the machineries are actually here. One, that is promoter. Two, that is structural gene. Three, that is terminator. What did I tell? I told for this transcription, for the process of transcription, I need the transcription unit. The machineries, what is going to be used? Number one is promoter, number two is structural gene, and number three is terminator. So here I am going to draw a strand, a double-stranded DNA, but I am going to separate both the strands, that easily I can show you what exactly happens at the time of transcription. So let me draw separated strand of DNA here. Let me make it very easy. 3' to 5'. So obviously we know DNA strands, double strands are anti-parallel. So obviously what will be the polarity of this? 5' to 3'. Now let me draw some bases. The bases are negligible. You no need to memorize for your examination. You just need to understand that. So I am here putting some bases. Let me make it a little bit narrow so easily we can write more. A, C, C, G, T, T, G, C, C, A, C. Okay, let me change one only. Here I am going to use A. Okay, so easily we can understand, once we check this strand, easily we can understand what are the codings present in the anti-parallel strand or the opposite strand. Okay, so here we are going to write exactly the opposite or complementary base pairing, that is from A. If it is A, so we have to write here T, then here it is G, here it is again G, here it is C, here it is A, then T, C, G, G, T, T, and G. So these are the strands. I have exactly separated both the strands. We know that DNA strands are intertwined, but still I have separated that for the use to make you understand easily. Okay, now if we talk about the structural gene, the structural gene is exactly the portion which is going to be transcribed into messenger RNA. Why the name is messenger RNA? Actually this RNA is carrying some information or message to produce the protein molecules. Okay, now here first if we talk about this, let me take it like that, so lavender color is very nice for this. Okay, so first if we talk about production of the mRNA, we need an enzyme. The enzyme is going to work on this DNA strand. So the enzyme's name will be DNA dependent. What is going to be produced? RNA. So what polymers are going to be produced? RNA. So DNA dependent RNA polymerase. This is the name of the enzyme what we are going to use here. So DNA dependent RNA polymerase, in short I am going to tell only RNA pol. It is going to start the transcription here, but easily it cannot start. There is a specific region where it has to initiate the process. This area is called as promoter. Here we need another factor which is called as sigma factor to initiate this transcription process. This is the sigma factor. Sigma factor. So So now in this way we can exactly start now the transcription process. Transcription process, what are the enzymes involved? Only one enzyme is involved, that is DNA dependent RNA polymerase. Now I am going to prepare my RNA strand here. RNA is a single stranded, right? RNA is a single stranded molecule. So the single strand, which strand it is going to take? First of all, we have to understand one is coding strand or antisense strand and another is sense strand. Please check first this structure. Okay, now what happens here? Just understand here. See, the RNA molecule I am going to prepare, that will be C, A. For A, it cannot be thymine. It should be always uracil because I am making the RNA molecule. So it will be U, then C, then G, G, again U. Okay, now it has to stop in a specific region. Where it has to stop? There is a specific region which is called as the terminator. So once the terminator region the enzyme reaches, there is a factor which is called as rho factor, and when it is attached to the enzyme stopped, it became inactive, and the mRNA which is produced, that will be completely separated from this strand. So now we have our mRNA here which is completely separated, a new strand. Now we have to understand one thing. I have written here 3' to 5'. So obviously RNA molecule which is produced, that is in the opposite direction. It is always just like replication, 5' to 3' direction. The RNA strand which is produced, again it has produced in 5' to 3' direction. Thank you.